Hi, my name is Keith. I'm a postdoctoral fellow in Dr. Xiaofeng Zhang's lab at the National Library of Medicine. We are interested in microbial genomics and evolution, especially in the context of human health. Over the course of our research, we've developed multiple tools that help us identify and analyze interesting features within microbial genomes, and I want to tell you about one of them today. With the rapid development of new and cheap sequencing approaches, there has been a boom in the number of genomes and metagenomes available from a huge range of environments. These genomes and metagenomes contain a wealth of information, but deriving biological insights from them can be challenging. When searching for genes, we often use multiple search approaches, relying on annotations from different databases, and then attempt to synthesize a consensus from these different results. This process can be challenging, and it makes performing searches like these difficult, especially on large datasets. Our tool ProkFunFind has been designed to fill in the gap between annotation and insight by providing a way to synthesize different annotations and search approaches into one cohesive approach. Our tool breaks down into three different components. Feature identification, which involves the definition of what is being searched for. The ProkFunAnnotate tool, which is a pipeline for running a set of commonly used annotation tools on a set of genomes that you're interested in. And the ProkFunFind tool, which is used to search for functions that are defined by the feature models. The feature identification process involves the definition of what you want to search for. This step typically starts with information gathering. You start with an interesting feature that you want to search for example, a metabolic pathway or an enzyme complex. You then gather as much information about this feature as possible. This can be from previous studies, new experiments, or from annotation databases. The goal here is to understand what the essential and non-essential components of your feature of interest are, and if there are any alternatives to the components that you've identified. The last step in this process is to develop a model of your feature of interest. This model is a hierarchical representation of your feature. In this example, we have a pathway that consists of two reactions. Each reaction can be associated with multiple heterogeneous search terms, including sequences, protein domains, and ortholog definitions. This structure also allows you to define which components are essential or non-essential to your function of interest, which provides additional flexibility when searching for different kinds of biological features. The next step is the ProkFunAnnotate tool. This is set up as a snake-make pipeline that will take a set of genomes and predict the genes in those genomes using Proka, and lastly run a set of commonly used annotation tools on those genomes, creating a standardized set of annotations that we are going to base our search off of. The tools that are currently included in this pipeline are InterproScan, CoFamScan, and Eggnog Mapper. Running this set of tools along with the prediction of the gene sequences provides us with a wealth of annotation information that we can use in the subsequent search steps. The feature model and annotated genomes are then fed into the ProkFunFind tool, which uses that annotation information to search for your defined feature in each of the annotated genomes. The putative hits returned from this search can be analyzed to look at different things, like if the features are encoded by genes that fall into distinct gene clusters, what the presence or absence of the features are in the different genomes being analyzed, and how this feature and the genes associated with it seem to have evolved over the course of the evolution of the organisms being studied. We have been utilizing our ProkFunFind framework as a component of multiple studies where we have investigated the distribution of human health relevant functions across the microbial tree of life. These studies include an analysis of histamine secretion in the human gut microbiome and the characterization of the equal production gene cluster in different bacterial genomes. We have found this tool to be extremely useful, facilitating searches for functions using heterogeneous queries against large collections of microbial genomes. As we continue development of the tool, we intend to include additional search strategies and annotation approaches, and add on additional features to help analyze the results, including visualization of different components such as gene clusters, and the addition of other analysis methods like building phylogenetic trees of some of the putative hits, or identifying additional genes that are present in the same gene clusters as your genes of interest. If you want to learn more about our tool, please find a link to the GitHub repository and the QR code on the bottom right of the screen. And if you want to check out our website to learn more about the exciting research being done in the Zhang Lab, please feel free to check out the link in the bottom left of the screen. Thank you.